We'll concentrate on science today, and I'm going to talk about something which is a bit futuristic, you may think, because I'm going to talk about the optimization of humans, and especially of human brains, which is a topic of quite an interest, if we can get the picture going, because most of you have not achieved your full human potential. I always give this beautiful example of Amalia Deutscher, who is 11 years old, composed a big opera, has a number of concerts, played beautifully piano and violin, and she is an absolute genius on the, well, order of Mozart. She is frequently called a new Mozart. So why are you not new Mozarts? This is something that we have to change, and for that we have to optimize the brain. But for that we have to get the computer going. Anyway, this word neurocognitive comes from neuro, which is because we have neurons in our head, and cognitive because cognition is, well, how we learn about the world, and we try to combine. And for those of you who don't know Polish, my last name Duch means a ghost or a spirit. So depending on your preference. I show this picture because it just shows that not always what you see is what is there. You can think that this ghost is moving. That's not true. This is just half a second of a blurred picture that you see. But your brain will kind of try to convince you that it is a moving object, right? Well, but that's not the topic of today. I'll talk about optimization of brain processes, and that seems to be something that lunatics talk about. The question first is, can we understand what is going on in the brain? You just hear over and over, we don't understand the brain, we know nothing about the brain. It's all lies, of course. But it's true that there are still lots of details that you need to know. So first thing, what are the brain and mind relation? How myself is related to what happens in the brain, what we can measure. Then I'll talk a bit about brain networks and the neurodynamics that happens in our brain, and neurons as a space in which that takes place. And then I'll talk about fingerprints of mental activity. This is what we try to find out. We try to understand what brains are doing and interpret that in the sense of our inner experiences, mental activity, and how this is related to the dynamic functions of brain networks, and this is what we investigate. And then I will tell you a bit about human enhancement and optimization of brain processes and the technologies that come. Actually, I've mentioned this paper because I've been thinking about that for more than 2 decades now. And only now we are kind of ready with technology. And after a lot of effort with the lab that we have to tackle this type of things. And I have a lot of other projects, actually, many of them unfinished. My problem is that every year I find something much more exciting than what I was doing last year. And so I leave things unfinished to my collaborators. So if you want to hear about computational creativity, insight, intuition, imagery from computational point of view, if you want to hear about computational intelligence, imagery agnosia, our work on infants, neurocognitive approach to language, and lots of other things, and autism and other things, I'll be happy to talk with you. And I also have a long kind of presentation that I gave a few months ago on machine learning algorithms that are worth developing. But I just left it to my former collaborators and students. So these are not the things I'm going to talk. I'm going to talk about what we're doing with my neurofanatics. These guys are actually, some of them are from psychology. There is one philosopher among us, very useful fellow, I must say. There are some physicists. There are some people doing signal processing, all kinds of things. And some hardware people. This guy on the right, for example, has a real proper hardware workshop. And myself, I started from physics. I worked in computer science or neural networks and machine learning for many years. And now sometimes I say I'm doing the diapers because the first big project we had in cognitive science was with infants. So well, my girls are doing the diapers, but that's still my area. And to do these things that we do, we need to have a lab. And the lab was very difficult to start from zero because we had no one who knew what to do. But finally, we have a lab with lots of toys, including the functional magnetic resonance, and I have 4 PhD students working in there. And we had to learn all this from scratch. And then we've made collaboration with people from the Institute of Neuroscience, more or less in Polish Academy of Science, which is Nensky Institute. We've made a collaboration with people in World Hearing Institute and have some big projects to search for brain cognitive activity. And if some of you are looking for a good postdoc position, I have one urgent need for a postdoc. So please talk to me about that. And unfortunately, it has to be about signal processing and maybe computational intelligence, etc. So more on technical side. We have some other people doing experiments, psychologists doing various things. So OK, we have 2 worlds, basically, as you know. One is the brain, and we can measure lots of things with the brain because the toys that we have allow us to do that. EEG, magnetic encephalography, the near-infrared techniques, the positron emission tomography, the functional MRI, many other techniques. Now, the labs that we have access to, we don't have NIRS yet, we don't have PET, but nearby labs have it, so we can collaborate with them. The goal is to map what happens in the brain, the states of the brain, into some kind of mental states, the state of the mind. So now the question is, how do we describe the mind? How do we describe what happens in our heads? That's very difficult, but mental states we can measure in many different ways, and then we can try to, well, kind of map it into brain states and vice versa. I've been teaching at the SWPS, which some of you know. It's a kind of a social psychology university in Poland, which is a private university. Some lectures on how the brain are processing information and something on computational neuropsychology, thinking about these things. So my idea that I thought about a quarter century ago was that we need such mapping, that what is objective, what we can measure, should be mapped into subjective things. Now, that subjective things, some psychologists like this idea of having mental space or psychological space in which we'll talk about things that we can interpret on subjective level, that we can perceive shapes, colors, motions, that we can have moral perceptions, that we perceive different aspects of music, etc. So you can imagine that we have kind of dimensions in which we try to describe our mental states. And then what we try to do is to find out how to map from what we measure to this kind of mental states. Now, the first thing that you find out if you talk with philosophers is that they would like to have phenomenological description. And they have failed doing that, as you know, in 1920s, because phenomenology as the branch of philosophy has completely fallen apart. They never provided any reasonable way of describing that. Now, there are good reasons for that, and Harblut and Schwitzgabel, that's a philosopher and kind of psychiatrist, I would say, have published a very nice book describing inner experience. And if you look at the Schwitzgabel's page, you find actually some books that he has written on that, showing why is it so difficult to describe our inner experience. But this is a talk on philosophy in itself. I won't follow that. It's still very interesting that description of our inner experience is so difficult. Okay. So I'll talk a bit about brain networks as a space for neurodynamics, and I'll try to make it rather general without going into details. But, well, some people ask, what is thought? What is mental state? Many processes are going on in your brain. You have almost 100 billion neurons in there with 100,000 billion connections between them. Lots of information flow all the time. One of the favorite approaches now is just to let you lie down and think about blue oysters, whatever, and just to see what's the information flow. This is called the resting state, right? And then you can find out what kind of networks are working and where the information flows in the brain. Many of these processes go on parallel. Just to stand, I have to really use a lot of my brain power to control my muscles and my behavior. Most of it is automatic. Some of it you never heard about. If I ask you what your ghrelin level at the moment, you know your ghrelin level at this? It's very important for your body, right? Oh, very bad for you. But your brain knows. You see, your brain knows lots of things that you don't know. And so we don't have access to many processes. To know what's going on in my head, I have to sometimes, well, interpret internally the information flow. Sometimes I can interpret my actions and see what I'm doing and try to understand myself. And sometimes I can just measure certain processes and tell you what's in your brain. And you'll see that we are at the, well, a threshold of a dream, which may be a nightmare, which allows us now to say what is going on in your brain better than you know it yourself, which is a very surprising conclusion. So the question is, if there are so many chaotic processes, we can perceive internally only the strongest one, the most unique one, because they have to distinguish themselves from chaos. And how to extract stable intentions and stable states from such chaotic behavior that we have all this information flow in the brain. That's what people doing brain-computer interfaces are trying to do. And it's not easy. So what happens in the brain? Well, we have lots of subnetworks that are tightly coupled and send information among themselves. For example, visual networks of all sorts, auditory networks, the motor networks, etc. We have connections, we have regions of interest which will focus on specific parts of the brain. And sometimes people ask, where is consciousness, where is intelligence? It's in the whole brain, it's in the process. This is not a thing. And the process is that you have to control and actually coordinate all these networks together to solve certain problems, to have certain experiences, right? So there is a control system, and lots of these networks like auditory, visual, subcortical networks that control movements, ventral attention, dorsal attention, salience that tells you what's important, what to select from all the stimuli that you get. The multi-task connectivity will reveal, when you investigate that, a flexible hubs that allow you to coordinate all this and interpret that internally. So all these networks now, we can extract from what brains are doing. The first thing is that we want to see what are the connections between different parts of the brain. And this is called the connectome, the structural connectivity. Physicists were wise enough to invent certain techniques to do this, like functional MRI. We can then say, okay, this part is projecting because there are long axons to this part of the brain. And then once we have a space in which mental events may take place, mental events will be implemented by activating and information flow on the network. So this is the space in which that happens. If you have a part of the space damaged because of the stroke, for example, or mechanically damaged, some of the function that we have will go away, right? So functional connectivity is then related to the fact that some of these parts connected may collaborate in some tasks, and sometimes they won't collaborate. And to do that, to find it, we have to first say, okay, what are the regions of interest? We have to do parcellation of the brain to have the nodes. Then we look at the signal, how active they are when we just lie down in the scanner and try to relax, and it's making buzz, buzz, bzz, bzz, and your head is blowing up. And so you have a signal, and then once you have a signal, you may say, okay, is there a correlation between these different areas? So you look at the correlation matrix, and you try to find out those strong correlation, because those weak ones will not be noticed. They're necessary in the brain to control homeostasis, lots of things, but it doesn't matter that much. You just have to look at the strongest correlations. Once you have them, then you know that this part and this part of the brain collaborate. And so you have the whole brain graph and try to simplify that and have a kind of a graph that shows which parts of the brain talk with which parts in which conditions, right? So you have a network. And the network approach now to understanding what brains are doing is really very important. So once you have a network, let's say you put all these brain regions on a kind of a circle. The top one is the front, the prefrontal cortex. Then you have a motor cortex. You have something which is called the insular cortex. And then down there in the bottom, you have the back part, the occipital cortex. And then you have the subcortical areas. And then you say, okay, where are the strongest connections? Now, you see that, for example, the occipital area on the left and right is strongly connected because your left and right part of the visual cortex is always collaborating. And then the top one are the highly unique connections. The bottom one are those which are well preserved in everyone. So what is well preserved? For example, the occipital cortex left and right, because they have to work together so many times. But also you see this insular cortex, which is this yellow, strongly connected, and then the parietal cortex, which is the green one, etc. So some of it is unique, some of it is strongly connected. Now, if you look then at people with some problems, like autism spectrum disorder people, you can look at all these connections. You can then select the strongest one. And you can find out, as Yahata has published, that a small number of abnormal brain connections will predict that the ASD, the autism spectrum disorder, is going to develop. So this is the 29 strongest connections, 16 strongest connections between 29 selected regions that somehow differ significantly between people who are going to develop ASD and those who are not. So you can find out that the structure of these connections and the functional connections is very strongly related to, well, our abilities, but also whether we have fairly normal cognition or not. So once again, we put it on a circle and we see, okay, this is the control, the top one, ASD. You see that many strong connections are missing, and it's well known actually, but it gives you a very unique connectome. And then you have something which is called tuberous sclerosis, very bad condition, where even more connections are missing. So in some cases you see that the brains have lots of missing connections. Your network is not properly built because of many things, because of genetics, because of lack of stimulation, because of some poisoning, whatever. You don't have an optimal network. So the goal that we have, the long-term goal, is how to optimize your network. We hope that if we optimize your network, your brain is going to work better, especially those children with ASD or other serious problems, okay? Now, this is something that psychiatrists have noticed some time ago, and they say, well, we have to look at genetics, we have to look at the biology, how the cells are working, we have to look at how the networks are working, how the whole system is working. So we need multi-level approach, and they call it multi-level neuropsychiatric phenomics. Phenomics means that it's a description, right? And then they say, okay, mental diseases, we just look at the symptoms and we try to classify them by looking at the symptoms, and that's a wrong thing to do. We never could come up with a good description of what is a mental problem. So maybe we have to look at the regulation of all these large-scale networks. For example, we can look at 5 networks. This is what they proposed and called the research domain criteria. National Institute of Mental Health is promoting that as a way of looking at mental problems of all sorts. You know that they had the books like DSM-5 and other books which just list the symptoms, and then psychiatrists will look at their objects and say, okay, this is the symptoms that we see, so probably this is kind of a disease that we can understand, classify, at least classify, and then try to use some therapeutic ways. But now they say, okay, let's look at the negative valence system. Fear, aversive situations, anxiety, loss, etc. Is it working correctly? Is it really responding correctly? Or positive valence system, or cognitive system. Each of the systems is very complex, right? Social processes system, arousal, regulatory system that keeps us aroused and going, okay? So they say, let's build a big matrix. For example, this is the cognitive domain, and you look at attention, you look at perception, visual, auditory, olfactory, you look at memory, you look at cognitive control processes, lots of these things. How do we understand these processes? We have to look at the genes because they influence molecules, which are the proteins building our cells, and then the proteins are building the cells, which then will connect and form microcircuits of all sorts, and then bigger areas, tissues, some parts of the brain, regions of interest that will actually show some physiological reactions. And then all that is actually coding behavior or allowing us to behave in a certain way. And in the end, we then relate that to self-reports. How do we feel about that situations and etc. So all this we have to understand. And this is, as you see, a whiteboard, terra incognita. This is like, 14th century exploration still. We know very little about how to feel this. So little that recently they've removed all this genetics from information because they are not certain about that. Okay. So step by step, we think that we can try to describe different large-scale networks. And the 5 networks that I showed you are not quite unique because you can have a positive affect, attention, etc. You can split them into more precise subnetworks. You can look at salience, negative effects, and then you can look also at default mode. When I don't do anything, okay, I see some kind of information flow between these regions of the brain. Okay, so this is how people now try to understand the behavior, looking at that, and then they look at different parts or regions of interest, that each row or each column is a region of interest, and they try to see how correlated they are with the others. So not just left and right brain, like in lots of popular press, not just front and maybe middle and brainstem, etc., but 1,000 of different regions that we want to understand how they collaborate to create our mental states and behavior. And the high correlation is this, the blue, the low correlation is the red one. And you see, for example, that visual system is well tightly correlated, but also with some motor system, because when you see something, you might want to touch it or act on this. So motor and visual behavior is very correlated. There is lots of blue in the top there. And then the salience is correlated with lots of things, etc. So we do this type of analysis. And then if we are careful, we can see, okay, one of these big networks like default mode network, when we look at shorter periods of time, you find that there are 9 different versions of it. And transitions between them are very important. Transitions allow us to say quite a bit about mental states of people too. Actually, this looking at the transition has been quite important recently, because people have tried to derive features like, okay, this part of the brain, can we interpret that? This is like phrenologists that you remember in 19th century have been very active, right? They just thought that okay, you can localize functions. You can localize just processes, networks. That's what we try to do. Okay, all this has been done with fMRI. fMRI tries to see how much oxygen is used by different parts of the brain. So how hard is it that let's say this brain region is working, and whether that correlates with other brain regions, etc. Okay, but fMRI is expensive. You need a machine which is like 40 tons and costs you 10 million zlotys or a few $1,000,000. You need a team of 6 people maybe to work on that, to interpret that. You need radiologists. This is very expensive, and it won't go down very quickly. It's developing, but not that quickly. What is exciting nowadays is that people doing analysis of EEG have shown that they can do measurements simultaneously in very high magnetic field using EEG. And my friend Bodurka, who studied with me and comes from the same high school, who is now a professor in US, has published the first paper showing how to do that, which made us quite happy. Again, there is lots of technology in the sense of signal processing behind that, but you can get signals which are called BOLT, and they are from fMRI, functional magnetic resonance, and you can get EEG signals, and you can compare them somehow. And surprisingly, you can find out that EEG allows you also to find very similar large-scale networks that you find from fMRI. And you see left column is EEG, fMRI, right is EEG, and you see a number of different subnetworks like this default mode network, the frontoparietal network, sensory motor networks. And these pictures look very similar if you just use EEG. So for the first time, actually, we know how to use simple technique. EEG is not that simple, really. We need really 128 electrodes, and we need to put them on the brain, etc. But we can do it much cheaper way, in much cheaper way than fMRI, okay? So people have found recently, Liu and other people, that you can extract information which looks like 14 different networks that also you can extract from fMRI just using EEG. So you have lots of these signals coming from high-density EEG, which is 128 electrodes or more. And I've just talked with people at UCL and learned that there is a machine which will have 1,500 electrodes, which I just can't imagine how it may work. How you can put 1,500 electrodes. They didn't want to talk about that, but this is something that they work on with one company that collaborates with them very closely. Okay, so you do lots of signal processing. That's why we need the postdoc. And in the end, you just try to see how the signals change in time during different tasks, etc., or during the period where you don't do anything. You have this default mode network. And then you just have electrode position. You just try to do co-registration with just normal magnetic resonance, not functional, to see to have an idea how the brain anatomy looks like, and then you can use the head model and have the information about the brain states. Okay, you can do it in different ways. For example, after 10 minutes of fMRI, you get the networks, and you can do similar things with EEG. So this is a great progress, I would say. And to do it good in a good way, we unfortunately need a lot of work which is on signal processing side, and that means that we have this 128 electrodes, and this changes in time. And some of it is very noisy because of different things. We need Faraday cage, which we just bought for 150,000 zlotys, and try to use that to shield us from external influence of electromagnetic fields, etc. And then we may have different signals, MEG, EEG, and fMRI. We want to integrate this information, so we have time, we have space because of the electrodes, we have a number of trials that we have, different conditions, different people. So we have multidimensional data. And this multidimensional data, we've learned how to analyze using the tensor approach or tensor techniques. And Andrzej Cichocki, who has been for 25 years at RIKEN Brain Science Institute. RIKEN is kind of Academy of Science in Japan, close to Tokyo, has invented a number of these techniques, and we are quite happy that he has joined our project and moved to our university because he had to retire in Japan finally, which is good for us. Okay, tensorization of convolutive deep learning networks. That's the technical part of it, which I won't talk about, but that gives us a chance and tools to analyze the signals in a better way and find out what you have in your head. So what are the fingerprints of mental activity? Okay, you think that you have a private life, and this is going to end very soon. Why? Because we've learned how to extract information about the images that you have in your head when you image things, when you dream, whatever. One way of doing it is that, okay, you see an image, and we try to extract features from this image. And you can do it many different ways. One way is to use the machine learning features, which are called here SIFT, GIST, HMAX, and other things like that. People doing analysis in computer science have learned how to extract different features from patches of images like this. So you have basically information about HMAX1 features. The initial is, well, not very strong, but then you have a group which is much stronger, etc. Okay, you analyze that, and you have thousands of these features, right? And then you look at the fMRI, and you show people different pictures, and you get your activity, like I showed you, which is the activity of different regions of the brain when you have seen the image, okay? Now, you use machine learning techniques like deep learning to decode that. So each image has a brain activity, and then you have a feature pattern that you extract. These features that we have learned how to do in kind of a computer science way, we try to extract them from the images that are there and correlate them with the brain activity and learn how to get them from the brain activity alone. So once you've done that for, let's say, a few hundred of pictures or something like that, you can show people quite different image, and you can extract the feature pattern, and this feature pattern you can fit to 100,000 different images that you find in Google, and you can say what you have in your mind, because you have a feature pattern. It's very generic. You just train that on a few hundred because you learn features. You don't learn correlation of images with fMRI. You learn just correlation of images with features that you derive from fMRI. As a result, you can dream, you can just imagine things, you can see things, and we can just look at your fMRI and say, okay, what do you have in your head? And as you know, sometimes dreams are very fuzzy and we cannot recall them, but if you record the signals from your brain, then you can extract that. Now, the challenge is, can we use the EEG signals, because it will be much easier to do the same? I hope we can, but I won't show you anything on that yet. I will show you something different. Now, you may say, okay, these images may be still just selected, but we can try to reconstruct them from the features. And reconstruction is still fairly fuzzy. Now, why is that? Because we look through the skull. We look through lots of tissue before we really see what happens at the neural level, especially with EEG. The signal is traveling through lots of complex tissue and the bone, etc. Now, imagine that you have electrodes in your brain. Okay, you have a macaque, because it's hard to find volunteers, although there are some people with epilepsy who have more than 200 electrodes in their brain nowadays, in the Mayo Clinic in States, for example. Okay, you have this macaque, and you have 205 electrodes in the brain of the macaque, and the macaque sees different faces. And then what you see in the electrodes is a series of impulses. So you learn to decode them in such a way that you can map the impulses that you see into features that allow you to see the faces. Now, the amazing thing is that out of the spikes of 205 electrodes, you hear sometimes that, brain is so complex, billions of neurons, you cannot understand them. This is all lies. 205 electrodes are enough to do something that you can see here. Maybe here. That is, this is the actual face. This is the face reconstructed from 205 electrodes and spikes that you measured. Which means if you have enough information directly from your brain, then you can actually see in quite details what you imagine or what you see or what is in your mind, right? Now, going back to this, DARPA, which is the Defense Agency, Advanced Research Program Agency, which is a part of Defense Ministry or department in US, has announced in 2016 a program where they want researchers to put 1 million nanowires in the brain of humans. Now, this is something that I could not imagine that is possible, but people doing nanotechnologies are doing very thin nanowires. And they say that 10% of them may be used actually to run currents through your brain, which will change the networks that are responsible for your inner world and your mind, right? So not only we can have 1 million electrodes and have a very detailed picture of what happens in your brain, much more detailed than I can know about it myself. Because what I know about my processes in the brain is this fuzzy internal kind of interpretation, right? And here I can really know lots of details that I will never know just from my own inner experience. And then I can also control the brains like that. This may happen within a few years' time because there are 6 or maybe 8 grants now and some progress in this. And maybe you've heard that Elon Musk, who is the Tesla director and also sends every 2 weeks now SpaceX into the space, has announced this program of neurolace. And that's still way off. I haven't heard about any results. Okay, but how do we reconstruct that? Because every time we think about something, there is a specific activity in the whole brain. And you see the strong activity here with the color and the weak activity with the blue color. And when you see, let's say, a murder scene or something like that, the parts of your brain that interpret what happened, for example, the parts that are visual, the parts that relate to auditory experiences, the part that relate to some specific motor activity, etc., or emotional, may actually be more active. And the specific activity allows you to build kind of maps for each concept that you think about and make a kind of atlas where you can find a specific part of the brain and say, okay, in which conditions, when you see a movie, when you hear the story, etc., this part of the brain is strongly active, and with which other parts it is correlated and works together. So this is an atlas that you can find yourself at the Gallant Lab at Berkeley. And it's been published in 2016. Actually, the first version was already 2012. And they have 1,700 different concepts. And you just go through semantic space and click on people, and people will be like Pope, policeman, or something like that. And you get specific activity of the brain that they have measured in 7 people. Average that there is some variance, of course, which is individual, etc. Now, building models of individual brains is going to be a big thing, and we know now roughly how to approach that. But that's, again, another thing. Now, if you listen to a story or you see a movie, etc., you may find out that there are new agents, new contexts, new situations, and this activity changes very quickly. Actually, this paper by Nicolaus Copernicus was reading stories activates neural representations of visual and motor experiences, but then she said it's like the movie editing room, that the things will resynchronize very quickly what happens in your brain when the scene is changing or where you think about other things. So you read the story and you see that causes, characters, objects, space, time, etc. will create a specific activity along the way, the chain of thoughts that are here. So what we found is important is that the transitions between different situations, even in a very rough way, when we look at EEG and just look which parts is more active and which is less active, like in this case, allow people to predict, for example, some conditions like schizophrenia, etc. Okay. So transitions are very important. We've done quite a lot of simulations which were of the sort of, let's say, trying to see how concepts are mapped into this map that I showed you, the semantics, where the semantics is basically each of these units there is coding some kind of microfeatures. So in reality, of course, these microfeatures are subnetworks in the network itself, but we show it, the names which are written or maybe phonologically spelled, and we look at how semantics is activated. And we try to understand the dynamics. And we see that once the word is shown, for some time you're staying within the same distribution, which means that you think about the concept. And then the system moves to another which is associated with this. It may be weakly associated. And then another. And after a few steps, it may come close to some that it has already seen. So we've looked at this dynamics from different perspectives. And actually, you can look at longer chains of thoughts, streams of thoughts. And actually, you can label each of these little boxes with a specific concept, and you can see how the system is learning. So all this brings us to this main goal of our big project, which is how to find the fingerprints of our mental activity from EEG, basically. And there are many technical ways of doing that. I've identified at least 8. I think right now I'm very excited about the virtual brain, which allows us to integrate EEG and neuroimaging data into models of individual brains. Recent paper has shown that if you have EEG information, you can predict what will happen in fMRI on a longer period, like 20 minutes. which is quite exciting and which really depends on the individual information about the brain. So I'm not going to talk about different ways, of course, because this will be very technical, but we'll just say a few words about recent experiments that we've been doing. We're interested not only in how the networks look like, but how they change and whether they are kind of static or when we solve problems, or whether they are dynamics. So some of you, if you've studied psychology, know that Daniel Kahneman has got a Nobel Prize, and he writes about the System 1 and System 2. The System 1 is, of course, the specialized processors, which are quite automatic, and System 2 is more flexible, which is this dynamic core that I showed you with all these subsystems around. So the questions we had here is that the whole brain network's properties during the performance may change if the performance required is getting more and more difficult. And there are lots of quantitative ways of evaluating the networks, like, is it modular? Can you really decompose that into some parts? Is the pass length between different parts a long one, many synapses on the way, or is it short? Is it, well, efficient? And there are different indices to measure the local efficiency and the global efficiency. Or how does that depend on the difficulty of the task or the cognitive load that we have? So all this, again, lots of technical details that I don't have time to talk about, but the experiments are fairly simple. You just use the classical Nbeck task experiment. Low cognitive effort is that you have to remember whether the new symbol, the new letter, is like the one that was just before, or the harder one is that you have to remember if it's like the one that you had 2 steps before, right? So you have to remember things. You have to remember that it was A, and then you have seen B, and then A appears, and you have to remember that it was A 2 steps back, and then D appears, and then you remember that it was not 2 steps back. So you just signal yes, no. Very simple experiment, and this experiment has been analyzed using parcellation into small number of nodes, which is the anatomical 90 different regions, like the gyruses in the brain, etc. Or there is a specific functional parcellation where we have 264 nodes. You look at the correlation matrices, you binarize them by looking at the thresholds, and depending on the thresholds, you get different matrices. And then you calculate all the things that you can calculate from networks like modularity, global efficiency, local efficiency, and other things like that. So you get out of it different ideas of how the cognitive load changes the functional connectivity or the functional networks that you see. And this was averaged over 35 participants. Maybe this is more clear. If you just look at local hubs, which are called provincial hubs, that is how strongly the system is working on the local level, then you see, of course, that there is visual activity and there is a prefrontal activity in the front. And in Tubeck case, you see that the local is not that important. You don't have a big, the colors are really regions that we have assigned to different types of networks, default mode network in particular. So if it's easy, lots of other things are going in your brain. It's almost like you don't do much, right? It's a default network. If it's hard, then the default network has to, well, switch off, and then executive processes have to be turned on. So in this case, you see that in more difficult cases, you have connector hubs which become much more important. So connector hubs are those regions of the brain that connect strongly with others which are far away, not in the same brain regions, basically. So dynamic change of the landscape of well, what happens in the brain, or where are the stable states, the attractors, is changing depending on the load. And well, we can relate that to modularity and efficiency and other things, which, okay, I'm not going to discuss that because there are too many effects related to it. Maybe we just look at the changes in the pass lengths, how many synapses and how long signal have to go, is related to intellectual performance. We are doing lots of experiments to find out how the intelligence is related to some of these parameters. Modularity is related to working memory performance, but also the capacity of working memory. And as you know, the working memory is very important because more you can remember and hold in your mind for a while, the better chance that you're going to find something associating important things, right? So psychologists know it quite well that working memory is very important for intelligence and capacity of working memory and performance, etc. Okay, so we do lots of things trying to find these brain fingerprints. Now, how that is related to what I started to talk about, that is human enhancement and optimization of your brain. Well, as you know, we are enhancing lots of things now. For example, vision. Google is going to sell you pretty soon a nice lens that will change the color depending on the sugar level in your blood. Maybe there will be people who would like to see the bacteria, which is a very good idea for medical doctors. Or if you have hearing problems, there is implant that more than 250,000 people now have cochlear implants, which send the auditory information directly to the brain, to the acoustic nerve. Or you may have actually a deep brain stimulator in case of serious problems. And quite recently, if you have a problem with memory, you may have a stimulator that goes into your hippocampus, and part of your brain may be replaced by electronics, which is done by a fellow working on my former university. Okay, so what we want to see in this enhancement is how different procedures act on our brains and whether the brains change. So, for example, you may try to find out how games will affect the functional connectivity. We've been doing experiments where people have been playing logical games or just shoot and killed games and trying to find out how the change in functional connectivity at the level that we could do, which was EEG at that time, changes the brain and how that is related to performance. And you see some changes which, again, I mean, these are details I'm not going to talk about, but that's one thing, right? You just do kind of brain games or whatever, and you try to see whether your brain has changed. Another is that you may have a direct connection between what happens in your brain and have some kind of a signal that then the signals tell you, okay, I'm relaxed, that's fine, right? Or I'm tense and something is going on in my mind. Maybe I have a pain state in my brain and I want to reduce that. Lots of different things. And then you change that into something that you see. So this is the external loop that allows you to understand what happens with you, right? And this is called neurofeedback, of course. And I must say that 40 years ago, I was a student and I was writing just to make some money to a popular journal called Przekrój in Poland. It still exists. And I wrote the first article on the neurofeedback, which is called Electronica and Stress. So it's a long time, and despite that, you still find that people are doing exactly the same as they were doing in 40 years ago. This is something that we're going to change now because of our better understanding of how to measure processes in the brain. Okay, so new forms based on our fingerprints are coming, and that's one of the goals of our project. Then, of course, you may try to use the signals to control maybe a bit better or to read a bit better our states and interpret them. And this is called the brain-computer interfaces. You want to, well, use EEG signals, for example, analyzing in many different ways, and there are lots of technical details, and then there's a decision process, okay? It says, I want to go left because I'm on a wheelchair and want to use my brain states to control the car, for example. I mean, lots of this you can hear nowadays, that people are using their brains to control things. Now, unfortunately, EEG, as I said, is a very fuzzy signal. We are just learning how to do it better. You can put on your cortex, actually, but unfortunately, you have to open your skull first. And then you put on your cortex a kind of a mesh of electrodes, and then you have electrocorticography. That is done usually with people who suffer from epileptic seizures, which are not really responding to pharmacological treatment. Or you can just put these microelectrodes, as I told you, which is the best way. So I recommend you to put 1 million electrodes in your brain, then you are going to learn a lot about yourself. Unfortunately, that's technology which is not really yet ready for mass market, but it will be. So anyway, BCI. BCI is really a great technique. People have spent lots of money on that. You can look at all kinds of paradigm. What you measure: invasive electrodes in your brain, non-invasive. You can process that in different ways. You can predict intentions, emotions, mental states. You can try to help people who have emotional problems. You can use it in games, rehabilitation, treatment, etc., especially the after stroke. And usually people just use EEG for that. And usually people use mental, emotional states and motor imagery. But you can try to use lots of different things. So European Union has put lots of money into these brain-computer interfaces. Over 50 million euro have been spent in last decade on this BCI. There is more than 100 companies that are involved in that, and some of these systems, well, can do something, but unfortunately it was difficult. Now, since about 2 years, we have the mobile brain-body imaging techniques, which record movements, eye movements, various biosignals in natural situations, including EEG. And there is this MOBI system, which is, well, used to model the human cognitive event-related brain dynamics. Again, lots of these things are coming. Some of this is from Scott making at UCSD. And then we have the brain-computer-brain interfaces, which allow us to change directly what is going on in our brains. And it's very interdisciplinary, lots of things like sensor development, experiment design, neurophysiology, psychological analysis, etc. Lots of people involved in this type of things. Okay, and then you can use it as an active system. That is, you just try to create some kind of a brain activity, like you try to focus, for example, or to relax or something, or to generate motor intention. Or you can be just reactive. That is, we just look at what happens in your brain when you are doing things, and it's involuntary, but you can read, for example, that you got interested, you've noticed something. Well, you didn't notice it consciously, but your brain has noticed that. And if your brain has noticed that, then we have to point it out to you. This is what the American Army is doing. You just look for an enemy and you don't see anything, but your brain says, oh, there was something there. So you go back and you try to look more carefully, right? This is what is actually used in practice. And then there is a passive BCI, which is automatic involuntary brain activity, like arousal, stress, workload, etc. And I just want to know about what happens in my brain, and then if I know it, I can try to change that. There is lots of work on different kind of brain-computer interfaces, lots of applications in different domains. Let's say people usually sell it for paralyzed people, but of course there are not that many of them who cannot really move their fingers a lot. But in restoration of functions after, let's say, problems with damaged cortex after stroke, and to enhance the vigilance, and lots of other applications that are coming, including, for example, finding interesting things in the internet. Many neuroimaging techniques that are coming or are in use, but people try to use them in BCI. Okay, so I may just mention that, for example, tracking eye movement to control technical systems has been used in many applications, and this is part of it. We use it in infant research. In the infant project that we have a project with people who are in coma, suffer for disorders of consciousness, we look at different things, but again, this is fairly technical, so I won't discuss that. The passive BCI times are used for mental state assessments, like neuromarketing, cognitive workload, neuroergonomics, etc. But what became important in recent years is this open loop adaptation. That is, that we measure specific brain states, and then it leads to some actions, and that provides the system the feedback to which is based on mental state assessment to correct the actions which we want to learn and improve. This is quite effective if you combine that with the direct brain stimulation, and this is the part that we have just started. So we have a brain-computer interface in closed loop, which means you discover certain signals in the brain, you are able to interpret them to some way, you encode information that should be put back in the brain to improve your actions, and then you stimulate your brain. For example, I try to learn how to play piano, and then I want to stimulate my cortex to have a better movement or more precisely timed movement, etc., by reading what happens in my brain and then putting it back into the system. So this brain stimulation is now getting quite big. There is transcranial magnetic stimulation, which means that you have some coil and you pump a lot of energy, 5,000 amperes and 3,000 volts in few microseconds, which means that you have created a pulse which is like 3 Tesla, like in a big machine that we have that is 40 tons, in a short time. And you repeat that. And then the electrode is going to be very hot because of the amount of energy you pump in this. That's one of the problems that we have. So one thing is this magnetic field. It's very good to increase the neuroplasticity, which means to increase the willingness of your brain to change your networks. And that's what we want to do, because we want to have a large neuroplasticity, and we want you to do some tasks which will actually allow you to optimize your brains. And the other is just running currents of our source, direct current stimulation. Many sub-techniques that have developed in recent years. This is what I talked about yesterday with people at UCL, because they're very good in that. So, well, you can also put this electrocorticography kind of meshes on your brain, which is very good. And then you can control yourself using your cell phone, for example. Very good idea if you are epileptic, if you suffer from obsessive compulsive disorder, if you have a phobia that you cannot really overcome, and it is really very hard in your life. Then you just, if you feel that, okay, I have to wash my hands again, and I've done that already 50 times, you just move a knob, a slider in your telephone, and it's gone. That's what is going to happen, and that's what is happening because people are doing the brain stimulation, and they develop materials which are now quite friendly. You can keep that for years, and nothing happens with your cortex, right? And before, you had to change it too quickly. Now, you have people with Parkinson's disease and lots of other things, and this is what our neurosurgeons are doing at our university also, because we have Collegium Medicum at the Nicolaus Copernicus University, where I come from, and also in this military hospital, they're pretty good neurosurgeons. New approaches are coming. The ultrasound interference, wonderful new technology. Very few papers so far, but it's coming in a big way now. So lots of these functions can be controlled if you have a deep brain stimulator. You have electrodes in your brain. And then you can also try to synchronize certain processes in your brain to make it more efficient. For example, if the process is difficult, then you find that your top of the head, the parietal areas and the frontal areas have to collaborate. And they're far apart, so there are long axons connecting them, and the information flow is not always very quick. So what do? You run oscillations on the parietal area and on the frontal area, and you try to synchronize the processes there, and you find that working memory improves. This is very nice. This is what people from UCL has done quite recently in a series of experiments showing that oscillations that you can synchronize using the alternative currents and the direct stimulation can help you to increase your working memory. That's very nice, right? We would like to have, it easy without really, wrecking our brains. We would like to have things like that. EEG and some of these red electrodes are just running currents. And from EEG, we try to understand what happens in the brain. And from running currents, we know which parts of the brain to activate so that the information flow will be more efficient. So we are just buying something like that, which is a direct current stimulation with 256 electrodes, and some of them are used to do the DCS, running the currents. Increasing neuroplasticity with magnetic field, then running currents just to increase the information flow. Okay, all this leads me to the last step, which is neurocognitive technologies. They're already on the market. You can actually have NeuroRelax, which is a kind of headphones you put on your head, and there is some EEG electrodes, and they find out whether you are relaxed, whether you are in the mood to listen to what kind of music, and then they find music that fits to your mental state. That's called Mellow Mind, and you can buy that. Okay, but you can also buy something that helps you if you are an athlete and you want to push heavy weights. And well, when you push heavy weights, you have to send, your brain has to send to your muscles a lot of, well, information to contract them. Now, that obviously depends on the activity, not only of how strong your muscles are, but also how strong the signals that go to muscles are. And the signals are generated by motor cortex. So let's just put some electrodes in your motor cortex, which is EEG, which finds out, okay, this is the moment I'm trying to push. And then you have some DCS electrodes, which says, okay, this is the moment I have to run more current through your brain so that your motor cortex is going to be more active and sending more energy to your muscles. And surprisingly, these guys are really improving, maybe even 10%. And well, that's not something that you can find out as a kind of illegal doping, right? It's not pharmacological, it's something that you cannot measure, right? They simply learn how to press harder. And then if you want to pay a lot of attention, and you are a military expert, for example, and you have to find things, okay, I have to focus. What does that mean? I have to run my prefrontal cortex processes that will tell my visual cortex to really be active because I have to focus on what I'm doing, right? And then you find after 10 minutes or maybe half an hour, you can't do this, you're tired, right? Why are you tired? Because your prefrontal cortex has to really work hard to activate your cortex, your visual cortex, auditory cortex, whatever, to pay attention. Now, why do I have to use my prefrontal cortex? I just run my currents through the cortex that has to be active. And if I do this, and you can buy something that's called Think, very primitive thing, right? But still, it's on the market. Well, depending whether you want to relax or you want to be attentive, it just runs some currents through your brain. And it helps to activate brain areas which are needed to perform the function. Okay, so all this is coming, and it would be very nice to have it for learning, right? So we want to have an interface that will help us to learn faster, and not just the motor things. And so, for example, in the recent paper, we see that people have used this for natural brain information interface, recommending information that has high relevance that is related to how your brain is reacting. Maybe I haven't noticed it's interesting, but my brain has noticed that. My brain is always much more wise than I am. Right? I'm making lots of stupid things, and my brain tries to correct me. And sometimes the vice versa, right? I consciously find out, okay, I've done something stupid, and that's because my brain has made me do it. So teaching skills by stimulating cortex is an interesting idea. If you have the electrodes, you can do microstimulation, which are too low to evoke muscle activations, but still they prepare your premotor cortex for complex activity, like playing piano. And there are first papers showing that you can inject instructions into premotor cortex, which will be fantastic if I could learn playing piano without really playing piano, right? Which is hard work to do, and which takes you many years, or dancing, or whatever you want to do. And this is what the military is doing in the US, of course. They have this engagement skills trainer. They don't play piano, they shoot people, as you know. And you are, well, shown in virtual reality some kind of a scenes, and an expert knows whom to shoot and who to save, right? And so you have to read the expert states of the brain, and then you have to activate the novice to do the same by activating the similar areas in the brain. This is what is called now the Intific NeuroEst, the Neuro Engagement Skills Trainer. Unfortunately, the army has most of the money, so it's a real life transfer learning. And this has been done in a very primitive way so far, but now we have systems that have more than 100 channels. We can be much more precise. And as I mentioned, UCL people told me that there is a system they work on. They don't know if it's going to be really working and useful, but 1,500 channels would be absolutely fantastic if that would be possible. Now, DARPA last year has announced another program, Enhanced Learning of a Wide Range of Cognitive Skills, which will be reducing the cost and duration of their extensive training regimen by increasing neuroplasticity. So many military projects that are related to how we can train people, especially train, but not only those that should, but they talk about foreign language specialists, they talk about intelligence analysts, cryptographers, and many others. So maybe general learning is also in their area. And finally, we have some ideas that people talk about quite frequently, saying maybe we can communicate just by thinking. If I can know what's in my brain, I can tell you that without really speaking, right? That's still a very far-fetched idea. I don't think this is going to be done soon. But this brain reading on the left side with EEG can, transmit simple motor intentions to the other fellow, who is then activated using DCS, or in this case with magnetic pulses with TMS, and then his hand is twitching, maybe right or left. And so you can send like the Morse code from one person to the other. One is just thinking, but it's slow. You spend lots of time, and then you see, sweet twitching of your fingers. That's all you can do. Now you can think, okay, this is too primitive, and it never will anything come out of it. But if you go to the Science Museum here in London, you find, okay, there was a telegraph, just Morse code, right? Just twitching. And then there was a radio, there was a TV, and now we have virtual reality. Pretty soon we will just be immersed in virtual reality and forget about reality, which is not interesting at all. Okay, this is what is going to happen sooner or later, right? It's just very primitive technology at the moment. So this is my conclusions, basically. Optimization of brain processes are going to be possible. It may take quite a while, of course, because we have to be sure that we know what we are doing, and we are not sure yet. First, we need to find the good methods to discover the brain fingerprints of cognitive activity, and this is our big project at the moment. But also many other groups are working on that. We have to learn how to map between brains and mental states. This is our main goal. And then brain reading and stimulation and understanding of neurodynamics of how these networks reconfigure, how they change. They're all the key to these brain-computer-brain interfaces for voluntary self-regulation of brain functions. And there will be numerous therapeutic applications. We have now a subgroup that works on specific types of pain, for example. There is a pain matrix in the brain, and we want to break that by specific direct activations of certain target areas, let's say. So the roadmap is that we look at the brain neuroimaging, we model the brain processes, we link it with mental models, and then we look at this closed loop brain-computer-brain interfaces for conscious control and brain optimization. And then one big thing that is coming that I don't have time to talk about is that this neuromorphic hardware is coming. And this is chips which are not computer chips in the normal sense. They're really models of parts of neural tissue. Lots of neurons which, well, collaborate with each other, do things in parallel way. The neuromorphic hardware, it's called neuromorphic because it's designed to be like the networks of our neurons, with complexity that will be comparable with human brain. The IBM Synapse project has been finished 2 years ago, and you are going to have these chips soon in Samsung telephones, which will be used to recognize images and do other things. And Google on the 8th of May has shown the, Google Now, which is a kind of an avatar or some kind of a system that advises you. And people who have seen that said that it's strikingly human. Things like that are coming thanks to some of this neuromorphic hardware that will allow us to construct new brain models, and deeper understanding of brain functions is coming from this model-based approach, and lots of practical applications again. And then you have all these far-fetched DARPA projects with million electrodes in your brain, Neuralace from Neuralink and other things like that, that will help us to integrate brains with artificial systems and change our ability to control ourselves, but also, unfortunately, give abilities to other people to control ourselves, which is a scary part, right? So I was joking that one day my wife is going to buy me a nice hat, a bit heavy, and then she will control me for her cell phone because there will be electrodes in this. So lots of new initiatives are possible. And as you know, European Union has now pledged to put 20 billion euros into artificial intelligence. And part of it is related to these kind of things. So it's not just formal artificial intelligence, but using this to analyze brain signals. Okay, we had lots of fun conferences in our place, including even things like the neural history of art, neurohistory of arts. We have mirror neurons, phantomology. We have Homo communicativus conferences. Neuromania is just taking part in June, so you're all welcome. And even theologians have been interested, and we had an interdisciplinary workshop with theologians from France and Austria, Spain and Pampeluna and then Innsbruck in Austria, and lots of other came from all around the world talking about the relation between soul and brain, what makes us really human. I think the conclusion is very clear, and there is proceedings that you can find out from this workshop, and I even brought an article for them. So, okay, that's all I wanted to say.
To ask, in your own opinion, what would you say is the best way to, for instance, find out what kind or what type of epilepsy does one have?
Oh, that's a very hard question. I have good friends who are experts on that, who know much more than I do. So I wouldn't dare to say that because there are so many different kinds of epilepsy, of course, and well, there are different techniques that people use to analyze EEG, and I'm not sure what's the best technique for that. So I'm not a medical doctor, right? I mean, things like that people frequently ask me, and I say, I'm sorry, I'm not a medical doctor. I'm not qualified to give you any advice on things like that. First, and second, I really don't work on that on an everyday basis, but I can, probably point you to some people who are much better than I am. But in UK, you have many, excellent experts on that. So I have a good friend, and my former PhD student is working now on things like that in Singapore. But again, I don't follow their work that closely to help with that. So, okay, so.